Introduction. Acetic acid bacteria AAB are gram-negative root shape, aggregates aerobic bacteria capable of converting ethanol to acetic acid. AAB is commonly found in nature because it can grow on the variety of substrate. The purpose of this study was to
methodology of fermentation of papaya. So in this experiment, we actually uses two types of media. What is the media? One of it is GYC, which stands for glucose is calcium carbonated, and also the second one is bromoceropropyl agar. So in this experiment, we actually use various type of material and also apparatus. I will list up some here. The most common one is the Bunsen burner, and also the alcohol, and the petri dish, the paraffin, and also the inoculum loop, and also we have. Hi, it's me again. So I'll show you guys how we did the bromoceropropyl agar and also the glucose is calcium carbonated, which stands for GYC. There'll be various of steps, so please pay attention. So the first step will be ethanol and also distilled water were mixed. Then we add in 1.5 gram of papaya. Of course, the papaya was washed with distilled water and also cut. Then we add into the mixture of ethanol and also distilled water. The enrichment culture were incubated at 37 degrees Celsius for 3 to 5 days. Then we also do the zero dilution. Hi, so right now I'll show you guys how my group and I have done our gram staining. There'll be also various of steps. Some of you guys have already done it, so you guys are also quite familiar with it. But please bear with us, let's focus. So the first step is 1 drop of saline was taken and also placed on a dry clean glass slide. With a sterilized loop, 1 colony of bacteria from the agar plate was taken and also smeared the bacteria into the slime of the slide. The smear was allowed to air dry and so the slide was passed slowly through the flame. The smear was...
Result parts. The single colonies will grow through streaking plate will incubate in 30 degrees Celsius in 2 days and undergoes gram staining to identify whether the acetic acid producing bacteria. There will be a clear zone in the streaking plate. Result part. Gram staining. Bacterial cells were gram stained. Morphology of cells was examined under light microscope. The Acetobacter changed the median color from green to yellow and then yellow to green, while Glucobacter changed the median color into yellow in 14 days at 30 degrees Celsius. From the gram staining, the bacteria in bromoceropropyl agar is gram-negative. As we get, the result is pink color and the shape of the bacteria is rose-shaped. When we observe under light microscope, we assume that the result is capable to our experiment as acetic acid producing bacteria is a gram-negative bacteria with rose shape.
For the discussion part, me and BADRUL AZIM will further discuss based on the results we obtained. In this experiment, we isolated the acid bacteria from papaya as our sample. So in the first attempt, we successfully isolate acetic acid bacteria from GYC agar or GYC media. And how we can identify that we successfully get the acetic acid bacteria is through the gram staining, as we can observe that is a pink colonies observed under the microscope and also observed based on the shape of the size and the size of the colony. But in the first attempt using the bromoceropropyl culture, we fail to observe there is a pink colony, and this is made due to several reasons, such as we do not follow the proper aseptic technique. Like we accidentally open a leak before we go to isolate the colonies, or all the isolation procedures have to be carried out under laminar flow, so to avoid the contamination from the air. And also we can observe the acetic acid bacteria. The microscopic morphology observation is through the gram staining circular bacillus or root-shaped bacteria. So this finding also agree with another several authors such as the Yamada and also the others. Also found that the morphological of the acetic bacteria is gram-negative or pink color colonies. Okay, so when we isolate the acetic bacteria from the GYC media, we can observe there's a clear zone form. So why there's a clear zone form is due to the production of the acid, which is have to show there's a halogen form. So the clear zone is formed when we take the dilution of the acetic acid bacteria with the dilution 10 of the -5, and when after incubated at 30 degrees Celsius, And for the bromosyrup purpose, it's used as an indicator for the isolation of acidic bacteria. When we use the bromosyrup purpose, it's because it is easily to observe there is a color changes. So before there is a bacteria on the bromosyrup purpose, the color of the agar is purple. But after there's acidic bacteria found on the agar, they will change color. This is due to the changes of the pH. So initially there's a purple color, but after there's become a yellow agar. So this can indicate there's a acidic bacteria is found on the agar. So we also can observe from our experiment there is a color change during for the agar. So this indicates there's a acid bacteria found in the agar.
My name is GEETANANTHINI, daughter of Mahalingam, and my metric number is J20A0443. So in conclusion, acetic acid bacteria were isolated and identified from fermented fruit in this study. The enrichment culture technique was shown to be effective in promoting the growth of acetic acid bacteria from fruits. In this experiment, two types of culture agar are used, which is bromo-purple agar and GYC agar. Acetic acid bacteria were isolated in GYC and bromo-purple culture success, and acetic acid bacteria will grow. Acetic acid bacteria are gram-negative aerobic bacteria that can oxidize ethanol to produce acetic acid. Fermented papaya produced the most yellow zone on bromo-purple agar and GYC agar. They were chosen to produce acetic acid. It was suggested that these fruit be ripe and suitable for the enrichment technique. The isolated were able to grow at 4 to 10% ethanol concentration and produce the highest acetic acid content, indication their suitability. In this study of fermented papaya, acetic acid bacteria were identified as gram-negative, rod-shaped, obligate aerobic bacteria with the ability to oxidize ethanol to acetic acid. Thank you.